This episode of Cell and Gene, the podcast, is brought to you in partnership with Thermo Fisher Scientific, a pioneer in the development of groundbreaking plasmid solutions. For labs seeking to scale up plasmid purification, Thermo Fisher recommends the Kingfisher Plasmid Pro Maxi Processor, a fully automated benchtop instrument with a press-and-go design that eliminates setup, centrifugation, and pipetting, reducing errors in sample preparation. Additionally, the Mag Max Pro high-throughput no-spin plasmid purification kit automates lower-volume plasmid DNA purification, saving time and enabling scalability. Trust Thermo Fisher to streamline and enhance your plasmid purification processes. Hello, listeners, and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and I'm happy to introduce my guest to you, KSQ Therapeutics Chief Scientific Officer, Micah Benson. Micah, hello, welcome to Cell and Gene, the podcast.
Hi, Erin.
Thanks for being here. All right, so I'd love to hear it from you personally. Talk to us about KSQ Therapeutics, your mission, your pipeline, all that good stuff.
Yeah, absolutely. First, really excited to be here and looking forward to talk to you more about KSQ. So KSQ was founded on the idea of industrializing genome-scale CRISPR-Cas9 functional genomic screens to identify the genome's top targets. And we call this approach CRISPRomics. We began by applying this approach to address one of the biggest challenges associated with cancer care today, and that is the refractory nature of patients with advanced solid tumors to the current treatment paradigms. So we sought to identify the genome's top targets using our CRISPRomics platform to try and address this unmet need. We wanted to apply this approach to map two discrete mechanisms. The first mechanism being, can we identify novel pathways which can directly kill cancer cells while sparing healthy tissue? And can we do so in a manner that can be biomarker enabled? So we established a oncology CRISPRomics platform to address this challenge. Through these efforts, we identified a protein called USP1, which is a deubiquitinase. And we found that inhibition of this pathway in cancer models led to remarkable preclinical activity in patient samples, which are deficient in homologous recombination. Now, in particular, we found that inhibition of this pathway led to synergy with PARP inhibition in BRCA mutant patient samples. So we sought to identify the genome's top targets using our CRISPRomics platform to map the target space of two discrete mechanisms. The first being, can we identify top targets which, upon inhibition, can selectively kill cancer cells while sparing healthy tissue? And can this be done in a biomarker-enabled manner? And this led us to discover a protein called USP1. This is a deubiquitinase we discovered to be a central node regulating the DNA damage and response pathways, or DDR. We found that inhibition of this pathway drove remarkable anti-tumor activity in preclinical cancer models deficient in homologous recombination, including BRCA mutant subtypes. These efforts also discovered other undisclosed targets involved in DDR. The second mechanism we mapped was, can we identify novel targets which can optimize the ability of the immune response to kill cancers? Now, these target insights have actually set the foundation for our current portfolio as it stands today. Our lead programs are based on our discoveries in T cells, where we used our CRISPRomics approach to answer the question, what are the top targets within the genome, as well as the top target combinations, which can best enhance the ability of T cells to kill solid tumor cells? From these insights, we generated CRISPR-Cas9 engineered ETIL, or ETIL, which from these insights, we generated CRISPR-Cas9 engineered TIL, or ETIL, which is a gene-edited autologous cell therapy for the treatment of solid tumors. Our lead programs here are first KSQ-001EX, which is a single-edited ETIL program where we are inactivating a gene called SOX1. Our second program is KSQ-004EX, which is a dual-edited ETIL program where we're activating both SOX1 together with Regnase-1. So please remind me to tell you what the EX stands for these product names, because it involves a certain and interesting manufacturing process that we established. So I'll get to that later on. I should add that the platform has been remarkably productive in its ability to discover new biology this has actually created many opportunities which have allowed us to expand the impact of the platform through partnership. For example, we have a broad strategic collaboration established with Takeda focused on the development of novel therapeutics against 2 T-cell targets that we've identified with our platform. We're also in the process of discovering new targets that can enable NK cell function, and we've recently expanded this collaboration to identify new targets that can allow the immune system to overcome the acquired resistance mechanisms that are co-opted by cancers to suppress the immune system and evade it. Our efforts in targeted therapy have also served as the foundation for partnering, where we outlicensed our early stage DDR programs to Ono, as well as our clinical stage KSQ 4279 program, which targets the USP1 protein to Roche last year. These partnering activities have allowed us to focus on advancing our ETIL portfolio, as well as, more importantly, have built value for patients based on the insights of our CRISPR-omics approach.
Excellent. Thank you so much for that detailed description of KSQ and what you've been working on. You mentioned CRISPR-omics, and that's where I kind of want to go next with that discussion. Talk to our listeners about the CRISPR-omics discovery platform. So I guess in more detail, what is it? And, what was lacking or what needed to be enhanced in genomics in the first place that drove KSQ to develop it?
So I think the question is, what does industrialized genome scale CRISPR-Cas9 screens mean? So we believe that the use of CRISPR, similar gene editing approaches, and its ability to serve as a tool to uncover biological truth will have just as much impact, possibly more, than the use of gene editing in a clinical setting as a therapeutic modality. It's probably the most powerful tool that biologists currently have in their arsenal. I think back to my own time at the bench when I was a grad student and a postdoc. I really wish I had this tool to be able to query the function of genes in the various cells that I was studying. And I'd probably place it up there at the same level of importance as DNA sequencing and PCR in terms of impact. So why is this? Well, let's say we have a particular cellular function, and it could be the ability of a cancer cell to indefinitely proliferate and survive, or it could be the ability of an immune cell, such as a T cell or a natural killer cell, to infiltrate a tumor and kill it. So what we do is we perform pooled CRISPR screens to map the targets and the pathways involved in regulating these various functions. We take a population of cell types under inquiry, and we introduce a library of single guide RNAs or guides using transduction in a way so that each cell contains at random one guide integrated within its genome. What we can then do is actually track that guide based on the overall sequence of the guide, or based off of the inclusion of a barcode that we incorporate into the transduction vector. We then introduce Cas9 into these cells, and we get a cell that is now edited. And we now have a population of gene-edited cells. Each cell contains one gene inactivated at random. We take this population of cells and we place it into a functional setting that is relevant to physiological disease. So, for example, in the case of a cancer cell, it could be the ability of that cancer cell to metastasize, to survive, to proliferate. For an immune cell, it again could be the ability of that immune cell to infiltrate, survive, and function in a in vivo physiologically relevant context. And this approach works certainly more generally than just these two examples. What this allows us to do is ask the question, across the entirety of the genome, 20,000 genes, what are the top targets within the genome which are able to control the particular function of that particular cell being queried? And what we've been able to do is actually generate a very large data set of genome-wide screens using this approach across hundreds of different cancer cell models, as well as immune cell types. And this includes mouse and human T cells, T regulatory cells, NK cells, both under homeostatic conditions, as well as in the context of cancers. And we think that our edge really, our ability to industrialize this really comes down to how we've built our ability to run these screens across many different cell types, and importantly, how we've been able to do this in a in vivo physiologically relevant setting. For example, kind of winding back the clock, we started these efforts back in 2016. And we started very simply. Let's do a single genome-wide screen in a cancer cell model. It was hard. We're very happy when we did it. Fast forward a year from there, back in 2017, we had completed our first in vivo T-cell genome-wide screen. And at the time, we actually think, and I think this is true, that this was the first time anyone had ever done a in vivo genome-wide functional genomic screen ever. I would say that in vivo screen took many dozens of exceptional scientists over many, many months to complete. Now you fast forward to where we stand today, and the engine is still cranking. It's still very productive. We recently completed a series of in vivo screens in the context of cancer that is still generating novel, very important insights that are driving our ability to understand how tumors, as well as cells that are trying to kill tumors, are interacting with each other. So we take all of this data, we integrate this data into a massive data set that we call a CRISPR-omics knowledge base. And what this allows us to do is take a gene, and we could type the gene symbol into this data set. At the press of a button, we have an understanding as to how this particular gene functions across all of these different cell types, across all of these different functions that we're now measuring. I'll give you an example. Let's say we have PD-1, a very well understood gene. It is a negative regulator in the ability of T cells to kill solid tumor cells, very well understood. We'll take the gene symbol for PD-1, which is PACD1. We type it into our CRISPR-omics knowledge base, and what we could see is that across hundreds of different cancer cell models, PD-1 has no impact. And this is very consistent with everything we know about PD-1 and its role in cancer cell survival, proliferation, et cetera. It makes sense. If we go to our immune cell data sets, we see that PD-1 also has no impact on in vitro T cell settings. So this is also consistent with what we know about PD-1. When we go in vivo, we see that PD-1 has no impact in models which are insensitive to inhibition by PD-1 in the context of tumor. This is also consistent. It's only when we go into models which we know are sensitive to inhibition of PD-1 where we see PD-1 strongly scored. So we have this profile across every single gene within the genome. Now, the only way we can actually do this to generate these sorts of insights is by generating these insights at scale, by industrializing this approach. That allows us to prioritize those targets, which, if we're successful in developing novel therapeutics against them, hopefully gives us the best possible chance of impacting human disease, such as the scenario I mentioned earlier in advanced solid tumors. Now, importantly, this data set also lets us know which targets not to work on. I should say that we want to work on the strongest, the best targets that are able to provide the strongest effect size at whatever gene we're trying to manipulate. What we don't want to do is go after targets which may not have a very strong effect size. So we naturally deprioritize those based on the profile within our data set. It also lets us know which targets may have an unacceptable therapeutic index profile. By virtue of running these screens in cancer cell models, we actually have a pretty good idea of the sense of lethality if we engage these particular targets or pathways across many different tissue types.
Excellent. Thank you for that. I want to talk a little bit about your two lead engineered tumor-infiltrating lymphocyte programs. And I think this will give you the opportunity to kind of explain the EX in what is KSQ 001EX and KSQ 004EX. I know you talked about that at the top of our discussion. So both of those are advancing toward clinical studies for the treatment of solid tumors. And I would love for you to share with us some information about each one and give us maybe some status updates about each program.
Yeah, absolutely. So we're generating all of these very interesting target insights when we were cranking our CRISPR-omics target discovery engine. Now, we noticed something interesting that was happening in the cell therapy field at around the same time. We noticed that cell therapies that were being positioned for the treatment of liquid tumors were very successful. We also noticed that cell therapies that were being positioned for the treatment for solid tumors really were not seeing the same level of success. There seemed to be a bifurcation between cell therapies for these various settings. And we thought that our CRISPR-Omics insights could help address some of what was going on. To take a step back, if you're a patient and you are refractory to all lines of therapy and your tumor expresses either CD19 or BCMA, you then receive a CAR T cell therapy, there's a very good chance that you're going to respond. Now, if you're a patient, you failed all of their lines of therapy, you have a solid tumor, and receive a cell therapy, there's actually a very good chance that you're not going to respond. So, and we think this is because of two major reasons. The first is the inherent nature of tumor antigen heterogeneity that is kind of a hallmark of solid tumors. We now know, and there's been a lot of very elegant work that has been generated where the immune system is able to recognize solid tumors, and it does so by recognizing essentially a compilation of private neoantigens together with a handful of shared antigens. Now, the problem is that there are very few good shared antigens across patients with solid tumors. There are, I think those that are good are very focused in the amount of patients that they can actually address. So the problem there is that patients tend to have a personalized neoantigen essentially pattern that would be the ideal antigens you'd want to develop an immune response against. Now, there's one approach that was out there that was actually effectively taking advantage of this, and this is TIL therapy, or tumor-infiltrating lymphocyte therapy. And what TIL therapy is essentially co-opting a patient's personalized immune response against cancer. TIL are extracted directly from the tumor microenvironment, and those TIL that are extracted contain a population of T lymphocytes which have infiltrated the tumor, are recognizing the tumor, and are attempting to kill that tumor. Now, those TIL, unfortunately, are not strong enough. They've been weakened by the biology of the tumor microenvironment. What TIL therapy does is it extracts those TIL, expands them into the number of billions outside of the patient, and then they're reinfused back into the patient. And the whole idea is to provide the patient with the benefit of strength and numbers approach. Upon infusion of billions of TIL back into a patient, they can overwhelm the tumor, and if everything goes right, the patient can be cured. So TIL is a very good potential solution to help solve the tumor antigen heterogeneity problem. We also liked TIL because it's able to provide polyclonal comprehensive coverage of all of the specificities needed to drive a patient into a CR. So TIL kind of checks the boxes of the first major problem, which is why we thought patients were not responding to cell therapies if they had a solid tumor. The second problem is where we think we can help. And this is the fact that why aren't the TIL initially strong enough to kill solid tumors? It's because they have been rendered functionally inert by the impact of the solid tumor microenvironment. We thought that our CRISPR-Omics insights, the targets that we've identified, have the ability to enhance, augment the anti-tumor activity of those TIL. So we saw an opportunity where we could come in with our target insights that we generated with our approach and essentially enhance the anti-tumor activity of TIL. Now, we think that where we can benefit patients with our target insights that we're applying to TIL, it is really through enhancing the activity of patients that currently don't respond to TIL. So this includes within melanoma. I think really intriguingly for us, being able to apply these target insights to TIL to patients outside of the melanoma space. There's a ton of unmet need out there that we think that we can address. Maybe what I'll do is talk to you next about KSQ-001EX. As I mentioned, this is a SOX1 edited CRISPR-Cas9 engineered TIL product. We identified SOX1 in our CRISPR screens as a top hit. We identified it in our in vivo models where inactivation of SOX1 led to a remarkable enhancement in the ability of CD8 T cells to infiltrate solid tumor cells across many different models. And we also were able to identify SOX1 as a target that enhanced the ability of TIL to expand in vitro manufacturing settings. So SOX1, at the time, we actually had a pretty decent understanding of the biology of the target. It was a fairly underappreciated target as an IO target, but we knew, we had a general sense as to how this target could be working within the context of adopted T-cell transfer therapies as well as TIL. It's a negative regulator of cytokine signals. SOX1 stands for suppressor of cytokine signaling. And importantly, it's a negative regulator of cytokines that are involved in the ability of T cells to survive, to differentiate, and to generate a good anti-tumor immune response. So these cytokines include cytokines like IL-2, IL-7, IL-12, IL-15, and IL-21. So what we did was we evaluated the impact of SOX1 inactivation to enhance T-cell anti-tumor activity in our preclinical animal models, including in TIL. What we found was that inactivation of SOX1 led to a marked enhancement in the ability of the T cells to engraft, even in the absence of any lymphodepleting conditioning in our preclinical studies. Inactivation of SOX1 is able to boost the induction of central memory of formation within the lymphoid tissues, as well as the overall persistence of these TILs in our models. And similar to how we identified SOX1 as a target in our screen, that also enhanced the ability of T cells to infiltrate into tumors. We found that it drove remodeling the tumor microenvironment, including jettisoning out suppressant population of T regular cells. And we identified that SOX1 acted as kind of a critical checkpoint, which restrained the differentiation of tumor-killing effector cells derived from stem-like memory cells within the tumor microenvironment. Some really cool new biology that we're able to uncover. We then, so we used our CRISPR-Omics platform to identify SOX1. We also used our CRISPR-Omics screening approaches to generate therapeutic grade guides. This is a really cool approach because I haven't seen this done before. We actually generated a library of guides against every possible guide to get with the against the SOX1 locus. And the reason why we did this is we wanted to find the most potent and the most clean therapeutic guide that we could then use for therapeutic use. So we generated a library of guides against the SOX1 gene, and we ran a functional screen, and we identified that the most functional guides targeted a particular protein domain within the SOX1 coding sequence. And we selected one of these guides as our actual guide that we ended up manufacturing SOX1 with. So we cleared our IND for KSQ 001EX late last year. We're actually currently dosing patients in our phase 1/2 trial. We're currently positioning KSQ 001EX for metastatic melanoma, head and neck squamous cell carcinoma, and non-small cell lung carcinoma. And we have enrolled as well as dosed our first patients. Now, one thing I also want to point out, we identified SOX1 using our CRISPR approach. We identified therapeutic grade guides using our CRISPR screening approach. We're also engineered TIL using CRISPR-Cas9. And so it kind of speaks to the power of gene editing in general. We're using gene editing to identify new targets. We're using gene editing as a way to actually perform the drug discovery process and how we identify novel guides. And we're also using gene editing to actually engineer our actual drug product, which is kind of cool. And it speaks to kind of the power of, well, CRISPR. So our second ETIL program is KSQ 004EX. And KSQ 004EX is a dual edit where we're activating the SOX1 gene, which is the same gene we're activating for KSQ 001EX, as well as a second gene that we uncovered in our CRISPR screens called regnase-1. And regnase-1 was a second target, which very consistently scored as one of the top targets within our screens. In fact, at the time that we discovered this target, back in the early days of the company, we're very excited for when this gene symbol came out. And our data sciences group kind of announced that this was a new, very strong hit, simply because we had never heard of it before. And it kind of spoke to one of our initial, I guess, fears kind of going into this endeavor that we were just going to discover PD-1 again and nothing else. That this is one of the first examples of us discovering a target which at the time really had not been implicated as an immuno-oncology target. I should add that this target has been since reported by other groups quite elegantly, using very similar approaches that we did. Um, when we conducted our first genome-wide screens, one of our lead scientists had a very good idea as to how we could take the next big leap forward, and asked the question as what the top target combinations might be. So we generated our genome-wide screens. We identified Regnase and SOX1. We took all of our top hits and we actually combined them at random with each other using an approach that we call CRISPR squared. And what we found was that the first time we ran the study, we actually cured all the mice. Just the presence of the library within T cells enhanced their anti-tumor activity strong enough that we started seeing pretty dramatic, actually, tumor regressions. So we had to rerun the study. And even then, after growing the tumors much larger, we were still seeing tumor regression. So we knew we had some pretty strong combinations within that library. So we looked at the CRISPR squared combination data, and we found that first, SOX1 was a fantastic combination partner. It combined well with a lot of our other top targets. In particular, we found that SOX1 combined quite strongly with the other top target we initially identified, which is Regnase-1. Inactivation of both of these targets led to around a 3,500-fold increase in the ability of these T cells to infiltrate and accumulate within tumors in comparison to unedited benchmark controls. So Regnase-1, it's an RNase. It's an enzyme that plays a crucial role in regulating the function of immune cells, including T cells, and it functions by targeting and degrading the 3' UTR of mRNAs encoding pro-inflammatory proteins, including cytokines, cell surface receptors, and components of the NF-kappa B pathway. And we think that dual inactivation of SOX1 and Regnase-1 by essentially making a cell hyper-responsive to cytokine signals, it tends to augment the ability of inactivation of Regnase to function, where we're seeing pretty interesting synergy between the SOX1 and the Regnase pathways with our preclinical animal models. And what this leads to is a pretty remarkable enhancement in the ability of T cells to kill solid tumors in our preclinical animal models. So hence why we selected this particular dual edit target combination for our KSQ 004EX product, which is currently in IND enabling studies. So now we'll get to what the EX actually stands for. EX stands for express. And it is referring to a next generation manufacturing process that we've optimized together with our manufacturing partners at CTMC for the manufacture of gene-edited engineered TIL. And the interesting thing about this manufacturing process is that it's 22 days. It's also feeder cell free. And what we're seeing is enhanced expansion of CD8 T cells. We're also seeing enhanced polyclonality of our ETIL drug product, and polyclonality is something that we want to see because we want to be able to retain all of the TCR specificities needed to drive patients into CRs.
Well, okay, thank you.
Okay, so as we know, solid tumors, major focus area. We also know that so too are autoimmune diseases. And so I'd love to hear from your perspective, the therapeutic potential for autoimmune disease. But perhaps maybe briefly talk us through the challenges and then maybe the opportunities as well to bring therapies to patients who have.
Yeah, absolutely. I mean, one of the reasons why I decided to join KSQ when the company was started was that I had just spent actually a number of years being quite active in the immunology and inflammation therapeutic area. And during my time working on autoimmunity, I saw that there was certainly a dire need for new targets in the autoimmune space, especially targets which engage new mechanisms as well as pathways. And these included emerging inflammatory pathways invoking both the innate immune system and non-immune cells in the treatment of chronic inflammatory diseases, as well as approaches which can invoke immune homeostasis with a possibility of even resetting the immune system. And so I think what we've seen over the past few years is some pretty encouraging, honestly, clinical reports of novel pathways being engaged, and which have been able to move the needle in a promising direction for patients. Just some examples that come to mind: OX40 pathway in derm, the CD40 ligand pathway in MS, And I think pretty interestingly is the use of the CD19 targeted cell therapies for B-cell autoimmune driven diseases. I think the exciting thing here is that B-cell depletion by virtue of an antibody has been evaluated clinically for years, even decades. For whatever reason, that just wasn't able to convey the same level of efficacy that the CD19 CAR T cell therapy had been able to convey. I think it's going to be very interesting to watch this space over the next few years to see if the ability of the CD19 CAR Ts to drive what looks like a true immunological reset in these patients is being driven simply by the depth of B-cell depletion that these CD19 CAR Ts are able to invoke, or a combination of B-cell depletion together with the upfront lymphodepleting chemotherapy that are currently administered to these patients. I think intriguingly, one of the exciting things about this observation is that it just kind of shows that there are things that you can do with a cell therapy that you really can't do with other modalities. So the fact that you're now seeing this pretty novel modality being able to generate some pretty amazing clinical results, kind of akin to the initial results that were seen in patient refractory B-cell lymphomas years ago when the CAR Ts first came out is pretty exciting. So we have been focusing our CRISPR-Omics platform to develop novel therapeutics for the treatment of cancer. We have also been, I think, quietly active in evaluating opportunities in autoimmune with our CRISPR screening approach. So as a reminder, you may recall that we positioned our CRISPR-Omics screening approach to identify and develop novel therapeutics for two mechanisms, one being directly killing of cancer cells, the second being enhancing the ability of the immune system to kill cancer cells. And that at face value seems pretty simplistic. If we think about the mechanisms at play within autoimmunity, I think one thing is for sure is that there's a whole lot more complexity in the mechanisms that are actually driving autoimmunity. Thinking about where we would want to best position our CRISPR-Omics screening approach to inhibit the immune system gets very complicated, very busy, very fast. And I think even within a particular autoimmune indication, there's a whole lot of complexity and heterogeneity within those patients, certainly with the mechanisms driving disease. So rather than investigate those mechanisms that inhibit the immune system directly, what we instead wanted to do was investigate mechanisms which can broadly boost the negative regulators of the immune system. And there's a very good cell type which you can do exactly that with, which is called the T regulatory cell. It's a type of T cell that is incredibly immunosuppressive, and it basically exists with the sole purpose of preventing our immune systems from attacking itself. It's been heavily implicated in autoimmune diseases, and I think there's been a lot of activity and excitement in trying to invoke ways to help boost the ability and the functionality of these T cells to help resolve autoimmune diseases.
Thank you. Finally, just talk us through briefly what's next for KSQ Therapeutics.
Yeah, so certainly focusing on continuing to bring our ETIL programs through clinical development. I think our aspiration here is to bring them to as many patients as possible. For KSQ-001-EX, as I mentioned, we are currently in phase 1. We have dosed our first patients. KSQ-004-EX, we are in late-stage IND-enabling studies. Now, we think that ETIL can be the rule for the treatment of cancer. However, we also think that there are going to be exceptions to that rule, and not all indications are going to be ripe for TIL. We think that, for example, in the treatment of CD19 and BCMA expressing liquid tumors, it probably does not make sense to develop a TIL therapy for those indications. So there's certainly exceptions. What we would like to do is think about opportunities where we can extend our SOX1, Regnase-1 target insights beyond TIL. So thinking about new opportunities, new types of T cell therapies that can be applied to help address some of those exceptions to the rule. So thinking about what new mechanism and opportunities exist even beyond oncology for CRISPR-Omics approach, certainly autoimmunity, as we briefly touched on, As mentioned, we've had some quiet progress in this space and believe our CRISPR-Omics approach can hopefully unlock some new and compelling biology here.
Excellent. Excellent. Okay. All right. Well, thank you so much, Micah, for all of this really great information and for your time today. And that wraps up this episode of Cell and Gene, the podcast. And for our listeners, please visit www.cellandgene.com and subscribe to this podcast. Tell your peers to do the same. And we will talk.